Yes, he is. He is still president until January 20th.
There's no Biden who
Will be the president. That is correct. But
Wait,
until the 20th, Donald Trump is still president.
is France in the U.S.?
France is not in the U.S. France is a different country.
Okay.
All right, we are live.
Then, Denver. I like waffles. Waffles are better than chocolate syrup and butter.
Oh, oh my God, it's right in front of me.
Chocolate syrup. What's better than green? Huh? What's better than green? Money?
Do we have people coming in?
We do. We have
Chocolate.
two so far, but it's still.
Better than money.
Oh yeah, it's not even started yet.
Yeah.
Okay, so we're fine.
Better than gold money? Gold dollars.
Did you guys get your candy?
Yeah.
Why is there an extra one here? Oh, you grabbed it from the bag. I got you. Who grabbed it from the bag? Raise your hand who grabbed it from the bag.
I did grab a piece of candy from the bag.
Notice how he won't raise his hand. It's very diplomatic of him.
I only grabbed one.
No, I understand that.
I grabbed them straight from the bag too. I can't stop.
Hey, Dad, do you have? Is that your second one or your first one? I just want to know.
Oh, it's number none yet.
Number two.
What's Nanya?
None your business.
Good morning, guys. Welcome, welcome. We are going to get started here within the next 5 minutes or so. Welcome to Mr. Ewald's class, Mr. Wen's class, and I think we even have Mrs. Becker's class in here for our 4th period live lecture. Make sure when you join the lecture that in the live chat you put your name as well as your teacher's name. And I'll put a little note in the live chat too. Make sure you have something to write on and something to write with as well, because you're going to be taking some notes and drawing some pictures and doing some different things as we move through the lecture. So again, we'll get started here in just a couple of minutes. So kind of get yourself ready, sit tight, and we'll be back with you here very shortly. Welcome, welcome. Happy Friday. Today is Friday. Let's do this. Oh yeah, we got a bunch coming in now. Yes.
I want to know what France's foods are. Well, I know a couple of them, but I don't know all. Do you know why I'm telling this out loud?
No, we have like rainy, gross, dreary Christmases. We need some snow.
Yeah. Dad, correct me. We were in Memphis for Christmas.
Yeah.
Correct or
Yeah,
incorrect?
that is correct.
Yeah. So we had no snow or just cold weather.
Yeah, it's just cold. Climate in Memphis is very similar to here, huh?
Yeah.
I would think.
Sorry.
I mean, we got a prototype.
Alrighty guys, so we're going to go ahead and get started here. So again, just to remind you, for those individuals that may have come in a little bit later, go ahead and take out a sheet of paper and something to write with to make sure to write down the notes and also write down the answers to the questions that we're going to be going over today. Okay? And by the way, happy Friday. Happy Friday.
Amen.
Hey, Mr. Ewald, why did the plant cell ask for a phone?
Oh, I don't know why.
Because he wanted to take a selfie.
But nice.
All right, so plasma membrane is the same thing as a cell membrane, and so don't want to make sure that we confuse you right off the bat. In terms of the plasma membrane, it is a thin, flexible boundary between the cell and the environment. And so the primary function, the main function of the cell membrane is to make sure to regulate movement of materials in and out of the cell. So we have to remember that our cells are living things. They need to make sure that they have materials. They need to make sure that they have enough stuff in order to survive. So things like oxygen, things like amino acids and lipids and proteins. And so we also have to make sure that they're able to get rid of things. And so that's what the function of the cell membrane is.
What is 12 shirts plus one tiny one?
All right, now, this idea of selective permeability. It's a really complicated word, but it's actually pretty simple. So this word permeability, it means the ability to be able to allow things to go in another thing. Okay? So in other words, we could have our cell membrane that is permeable allow certain things to come in. Now, in terms of selective, those things that come in, the cell membrane has an ability to control it. Okay? So it's very, very similar to, let's say, for instance, a coffee filter. You do not want any coffee grounds in your coffee. Okay? And so instead, you put that filter on, and it filters the water and not the coffee grounds. And so or it filters the coffee grounds, not the water, and so the water goes through the filter. Now, in terms of selective permeability, again, that plasma membrane, that cell membrane, controls the movement of those substances. And we've already talked about those substances that we want to make sure to have that come in, and also things that want to go out. So things that want to come in. We want oxygen to come in. We want CO2 to go out. We want nutrients to come in. We want waste to go out. So the plasma membrane is composed of the phospholipid bilayer. Now let's go ahead and break down that word bilayer. Okay, so if you remember, the prefix bi means two. And so we have two layers of phospholipids. And what is a phospholipid? It is a molecule that's made up of a polar phosphate head and a nonpolar fatty acid tail. So that's what makes up our cell membranes. And so the outside of that phospholipid bilayer is made up of those polar phosphate heads, and the inside is made up of those nonpolar fatty acid tails. All right, so in my class, whenever I talked about the phospholipid molecule, I referred to it as the jellyfish-like substance, okay? So the one with the polar phosphate head with the tentacles representing the non-polar fatty acid tail. So that's exactly what I'm talking about whenever I'm talking about the molecules that make up the cell membrane. Now, in terms of how the cell membrane behaves, we have an idea of how it behaves, the fluid mosaic model. Now, those phosphates, those polar phosphate heads, those fatty acid tails, those phospholipids will actually sit on the cell membrane, and it has the ability to move. It has the ability to adapt to the environment because the cell is not in a static environment. It is in a dynamic environment. So in other words, the environment is always changing. The cell needs to be able to adapt. That's one of the characteristics of living things, the ability to adapt. So if you cannot adapt, then you die. And so our cells are able to adapt. And so in this case, those phospholipids will change their shape depending on whatever needs it needs to meet in order for the cell to be able to survive. Now, along the lines of the mosaic, okay, so the mosaic is going to be the idea that we have all these different molecules on the cell membrane. Those molecules are going to help the cell membrane be able to do all sorts of different types of functions. So what we're going to focus on for this lesson are going to be about proteins and why proteins are important in making sure that the cell is able to achieve homeostasis, able to get its nutrients necessary, able to get rid of its waste. Okay? So again, fluid, the ability of the cell to be able to constantly move, constantly change, constantly adapt. The idea of mosaic, those different molecules that are embedded in and on the cell membrane. All right, so passive transport. So we need to be able to have those different types of substances move into the cell. And so we do this by accomplishing this type of transport, and that's going to be passive transport. So passive transport is the movement of particles across the cell membrane without using energy. I want you to underline this. I want you to star it. I want you to do whatever you need to do in order to remember that passive transport is transport without using energy. I want you to think about this example, okay? So imagine that you're on the Ohio River. And we're not going to swim the Ohio River because that's just disgusting, okay? But instead, we're going to get a canoe, okay? Whether you own a canoe, whether you rent a canoe, we're going to get a canoe. We're going to put it on the river. And then we're going to go downstream. Is that going to require energy in order to do that? No, it's not going to require energy because that current is just going to take that canoe and it's going to go with the flow. It's going to go with the flow. So that's the idea of passive transport. It goes with the flow. In terms of the three modes of passive transport, we have diffusion, facilitated diffusion, and osmosis. And we're going to talk about those later on. All right, now, so the first one we're going to talk about is diffusion. It is the movement of particles from an area of high concentration, to an area of lower concentration. So, I have a quick demonstration for you in terms of this diffusion. So we have this cup of water, okay? And in terms of the cup of water, we have all of these different food coloring. So I want you to choose a color for us so that we can show you what that diffusion looks like, okay? Do you want us to choose the blue color, the red color, the yellow color, or the green color? Okay? So in chat, I want you to tell us which color you want me to use for this demonstration. Now, as you guys are typing that in, I want you to also think about an example as well. So just imagine that you have that person that sprays the Body Axe or sprays that perfume. So where is that area of higher concentration going to be? It's going to be around that person, right, that sprayed it. But then what happens over time? All of a sudden you start smelling their Body Axe and you start smelling their perfume. That's diffusion because, again, it's going to spread evenly throughout that particular area.
All right, Mr. Wen. So we have a lot of colors coming in here. We had Miss Becker right off the bat vote for blue. We had a green. We got a blue for life. A bunch of blues. We do have some reds mixed in there. Couple reds, but I would say mostly we're talking about some Highlands blue, some UK blue.
No. We talking about Memphis blue here. Memphis blue, Memphis, home of the blues. Okay, so that's what we're going to use. We're going to use blue. Alrighty, so we have this container. We have this food coloring. So we're going to drop a few drops. And again, area of higher concentration. Those drops that are now diffusing in that cup. Even without me doing anything at all, you can start to see that it's spreading to the areas of lower concentration. Okay, now, you could increase the speed of diffusion. Okay, you can increase it by speeding up the molecules, either through temperature or also through pressure as well. Okay, so I'm going to do that. I'm going to speed up the rate of diffusion, and you're going to see that all of a sudden now, it diffuses evenly in that container. It does not separate out. And so now we have what we call a homogeneous mixture.
Look at that beautiful Highlands blue, UK blue cup.
All right, so in terms of this example, that's exactly what we see. We see in the beginning the separation of those two colors. Over time, those colors will come together and then form that homogeneous mixture, that mixture that looks exactly the same all throughout. All right, dynamic equilibrium. So that's exactly what we just achieved. We achieved dynamic equilibrium because we have materials that are evenly spaced out. Okay? So that's what the cell wants as well. It wants substances evenly spaced out inside and outside the cell. So reached when diffusion of material into the cell equals diffusion of material out of the cell. Now, that doesn't mean that movement stops. So movement will continue to go on and on and on in order to make sure to maintain that concentration.
Really hit the Memphis blue hard today. Going forward.
Alrighty, so our first question, what you're going to do is you're going to draw this membrane, drawing arrows to show movement, and what would happen over time for this cell to reach equilibrium. So here's the membrane, and you can see that there is holes or pores that are on this membrane. So what do you think is going to happen over time? All right, so if you are in Mrs. Becker's class, I would like you to go ahead and put the answer in chat. What do you think is going to happen over time in order for this cell to reach equilibrium?
All right, again, Mrs. Becker's class, you guys are on the spot. Question number 1. In the live chat, go ahead and put what you think would happen over time for this cell to reach equilibrium. So give us a nice little description of what you think might happen. All right, so we got some answers from Mrs. Becker's class coming in here. We got the molecules from the left side will diffuse onto the right side until they are equal. 3 of the molecules are going to go to the other side of the line so that they're equal on each side. When the concentration is the same throughout, the molecules will evenly diffuse from the left side into the right. They will diffuse, equal number of molecules on the right side. All particles will distribute and spread across the picture to reach equilibrium.
Awesome. Good job, guys. Okay, and so in order to demonstrate that, okay, so you're absolutely right. You're going to have molecules go from the left side to the right side because this is the area of higher concentration. This is the area of lower concentration. And so we're going to have these particular molecules go on the right side, these particular molecules go on the right side, and these particular molecules go on the right side. And so over time, okay, you're going to have those molecules that are going to be on that right side, and then these will not be there. Good job, guys, in terms of being able to show diffusion. All right, so for this one, same idea. Draw this membrane, drawing arrows to show movement. What would happen over time for this cell to reach equilibrium? Okay, so let's have my class put in the answers. Okay, so what do you think is going to happen over time in order to make sure that this cell is able to reach equilibrium?
All right, Mr. Wen's class, you guys are on. Go ahead and put your description of what would happen over time for this cell to reach equilibrium in the live chat. Let's see what you guys got. All right, so we're starting to get some answers in here from Mr. Wen's class. All right, so we have half of the particles would transfer to the empty side. 2 out of the 4 particles would move to the lower concentration side. Half would go to the lower concentration from the higher. Same thing. The cells will distribute equally to the right, the lower concentration side. 2 molecules would move to the lower concentration. 2 of the cells will move to the right side, creating equilibrium. Half will move to the lower concentration side. Half of the particles would go to the right.
Alrighty, guys, so here's what we have here. So we have this molecule that's going on the right side, this molecule that's going on the right side. Boom, we have equilibrium.
Whoa, 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 Mr. Memphis Blue. We can't do just that, right? So I see a big problem right off the bat, and what I see is the space in the membrane is only this big, but we have these huge particles over here on the left side. So I don't know about you, but for me, I'm not thinking that those can just pass right through the cell membrane. What do you think about that?
I think you're right. I think you're right. So they're not going to pass through, and so you're going to have 0 cells on the right side of that membrane. Okay, so 0 cells. So in other words, either this cell is already in equilibrium or it needs some help.
So let me throw something out there really quickly just to call somebody out. Gavin, I think from your class, mentioned something about the pores in the membrane. So he was thinking potentially there was going to be an issue with those pores. He mentioned that the pores would widen, so he's on the right track there for where we're going with
All
this.
right.
So I just wanted to throw that out there.
All right, so good job, Gavin, in terms of thinking about it the right way. Okay, so in terms of diffusion and the two things that we've seen. So we've seen that the smaller molecules are able to go across the cell membrane. So you can see that that cell membrane, again, there is a little bit of flexibility in allowing for that change in shape to allow for those molecules to diffuse across. Okay, so these smaller molecules are able to go through. However, these larger molecules are not able to go through because there's just not enough space, regardless of how they move. There's just not enough space. Okay, and so in terms of being able to get those across, there's another mechanism that we have to learn. And that's going to be facilitated diffusion. So in terms of facilitated diffusion, that is the movement of materials across the plasma membrane using proteins. Right there, using proteins. That is going to be what I want you to make sure to underline, star, circle, whatever the case may be. So that is really important in making sure that we're able to get those substances across. So based on the pictures, you can see that these proteins are called channel proteins, they're carrier proteins. The general term that I use are just membrane proteins. Okay? So those channel proteins, carrier proteins, membrane proteins, they allow for larger substances to move across. So again, we're trying to maintain that dynamic equilibrium. And so we're going from areas of high concentration to areas of low concentration, number one. But number two, it acts as a bridge to allow for those larger substances to move across. Because again, it cannot move across using just the cell membrane alone. Alrighty, so here's what you're going to add to your paper. Two differences between facilitated diffusion and simple diffusion. Two similarities between facilitated diffusion and simple diffusion. Okay, so let's have Mr. Ewald's class now. What I would like you to do in the chat is to give me just one difference between facilitated diffusion and simple diffusion, and just one similarity between facilitated diffusion and simple diffusion. I like your skin. I like your kitty cat. That's a good dog. Oh, Elsie, look at this. Something's wrong with this. Correct. Why don't you go ahead and correct that? Okay.
All right, so Mr. Ewald's class, I know you guys are out there in the live chat. Go ahead and throw out one similarity and one difference between facilitated diffusion and simple diffusion. So when we think about those two different mechanisms for passive transport, what's one similarity and one difference between facilitated diffusion and simple diffusion? I know you guys are out there. Let's go ahead and see those answers. All right, so we're starting to have a couple of answers come through. Major difference, instead of them just moving across the membrane to create equilibrium, the materials have to be transported using proteins. The smaller ones can go through the membrane, but the bigger ones need help. And then for a similarity, they both diffuse, right? So both of those materials are moving from high to low. Similarities, they're both transporting the materials across the plasma membrane. Difference, in simple diffusion, the molecules can pass directly across the membrane. One has to go through a longer process that's more complicated, so we have our proteins. They both end with an equal amount of particles on each side. Similarity, both diffuse. They both move particles, but one uses proteins. Similarity, both want to result in equilibrium. Awesome, awesome, awesome answers, guys. Wonderful.
Alrighty, guys. So again, those two differences between facilitated diffusion and simple diffusion, we'll go ahead and go over those. So, in terms of facilitated diffusion and simple diffusion, absolutely it has to do with those proteins. So those membrane proteins that we see in facilitated diffusion that we don't see in simple diffusion. And then the other one is going to be what you guys also allude to as well, the size of the particles. So facilitated diffusion is larger substances, whereas simple diffusion are smaller substances. In terms of your two similarities, so facilitated diffusion and simple diffusion, you're absolutely right. It goes from an area of high concentration to an area of lower concentration. But also, remember, because it's going from area high to low, it's not going to require any energy. Okay? The river on a boat, or the
Boat on a river?
Boat on a river.
There
Boat
you
on
go.
a river.
Passive transport, guys. Again, Mr. Wen mentioned this. I saw a little bit of confusion in the live chat. Passive transport means no energy. You did. So I'm just backing up what you said. You absolutely said that. Did I say they don't require energy? No, no. You said they don't require energy. I was just backing because somebody put something in the chat. So good stuff.
All right, so in terms of this example, draw this membrane drawing arrows to show movement. What would happen over time for this cell to reach equilibrium? Okay, so why don't you go ahead and draw this membrane, and show me what would happen over time for the cell to reach equilibrium.
You were more you made it more obvious that you don't want to swim. Yeah. All right, let's do everybody. Mr. Wen, Mr. Ewald, Mrs. Becker's class. Let's do everyone. Last names M through Z. Last names M through Z. Put those descriptions in the live chat. I know we're behind a little bit there, but last names M through Z. Everybody put those descriptions in the live chat. We'll take just a minute to wait for those to roll in, and then we'll go from there. So what are we going to see? What's going to happen over time for this cell to reach equilibrium?
Alrighty, so guys, in terms of what somebody else had said about those pores, you're absolutely right to think about those pores again. So what we have is a membrane protein. And so what we're going to do is we're going to make sure that these molecules will move across the membrane protein because they're larger substances. So we're going to go through this way, and we're going to go through this way. Okay? This would be a no. That would be bad because that doesn't fit. So that will not happen. So at this point in time, this would reach dynamic equilibrium because of the membrane protein and the fact that this is facilitated diffusion. If it helps you to remember, facilitate means to help. Okay? So if you can remember that facilitate means to help, you need to have that membrane protein to help with this transport.
Absolutely, and we have some answers coming in on the live chat. I just wanted to get in there. Lots of people saying that 2 of the particles can now move through the protein to the other side. So absolutely spot on with what we're looking at here for facilitated diffusion. Great stuff, guys.
Alrighty, so the last passive transport we're going to talk about is osmosis. And so in terms of diffusion of water, this is what we want to make sure that we focus on. This is about water. Okay, it's not about anything else but water. What are we talking about?
Water.
We're talking about water. Okay, so in this case, diffusion of water across a selectively permeable membrane. Remember, that selectively permeable means that it's able to choose to filter out some things that it doesn't need in order to make sure that it gets that water across. Alrighty, so we have three types of solutions that we want to talk about today. We want to talk about isotonic, hypotonic, and hypertonic. Now we want to focus on those prefixes because these words sound complicated, but they're actually not. So let's go ahead and break it down. Isotonic, hypotonic, and hypertonic. We can take those prefixes, iso, hypo, and hyper. Now iso may be a prefix that you've seen before in biology. So we've talked about isomers. I've talked about isometric exercises. Those are going to be using that prefix same. An isomer has the same chemical formula. Isometric exercises focus on the same muscle groups. And so iso means the same. So that solution is going to have the same of something. And we're going to see that here in just a second. Okay? In terms of hypo, okay, you might have heard the terms hypothermia or hypodermic needle. So in this case, whenever we're talking about hypothermia, that's when your body's internal temperatures goes way below than what it should be. When we're talking about a hypodermic needle, that's a needle that goes below the epidermis. And then in terms of hypertonic, hyper is an example of that would be hyperactive. Okay, so you know you got some classmates that are a little too hyperactive. So that means that their activity level is above the normal conditions of what that environment calls for. So that hyper means above. So our first solution is your isotonic solution. Water and dissolved substances diffuse into and out of the cell at the same rate. What that means is that water will go in and will go out of that cell in order to make sure, again, that we're maintaining that balance, maintaining that homeostasis. And that's due to the fact that the concentration inside the cell is the same as the concentration outside the cell. Okay? So the fact that they're the same means that that water is going to diffuse across at the same rate. All right. Hypotonic solutions. Solute concentration is higher inside the cell. Okay? So higher inside the cell. Water diffuses into the cell, and here is the golden rule I want you guys to remember. If there's anything in this lesson about osmosis that I want you to remember, it is this. Water will always go where there is more solute. Water will always go where there's more solute. Okay? So in this case, in a hypotonic solution, water is coming into the cell. And the reason why, again, is that solute concentration, these orange dots here. There's less orange dots on the outside than inside. Inside, there's a lot of orange dots. So that means that the concentration is really high inside that cell. So water it will want to go into that particular cell. Alrighty, and the last one is going to be hypertonic. So solute concentration is higher outside the cell. Okay, so outside the cell. Water diffuses out of the cell. Water will always go where there is more solute. So again, that golden rule that we want you to make sure that you remember. So in this case, in this cell, you can see the orange dots. There's very few orange dots inside that cell, but there's a lot of orange dots on the outside of the cell. That means the concentration is really high on the outside, so water will want to go out of the cell.
Mr. Wen, let's just say that you are stranded on a raft out in the ocean. You don't have access to any water. You're getting really thirsty, so you think that it's going to be a good idea for you to just start drinking a whole bunch of water right out of the ocean.
Oh yeah, I'm going to drink that ocean water.
I mean, you're thirsty. You're going to drink that ocean water.
Absolutely.
Why might it be a bad idea for you to drink some ocean water?
I mean, it tastes bad.
True. Why else?
Fish pee in it.
It could be. Maybe some poo.
All right, but I think it's because of the amount of salt that is inside the ocean.
Absolutely.
And so since there's a lot of salt inside the ocean, what your cells will want to do once you ingest that ocean water is that water is going to come out of your cells. All of a sudden now, you're thirstier than you were before. Okay, so again, life lesson, don't drink ocean water.
Yeah.
All right, question 5. Draw the picture, draw arrows to predict the movement of water. So here's what I want you to make sure to do. I want you to make sure to focus on this solute concentration, this water concentration that's outside the cell, and also the solute and water concentration that's inside the cell. Not only that, what I want you to do is tell me, is this a hypertonic solution, a hypotonic solution, or an isotonic solution? Okay? So if you are in, let's say all of the classes, so all the classes, if you are a January to a March baby, January to a March baby, okay, so January, February, March, okay, 3 months. So in all the classes, I want you to tell me about the movement of water. Will it go into the cell or will it go out of the cell? And then also tell me what type of solution it would be. Would it be a hypertonic solution, a hypotonic solution, or an isotonic solution?
All right. January to March birthdays. Put your descriptions in the chat. What is going to be your prediction for the movement of water based on the picture that you see up there? What's going to happen? And then what kind of solution do we have? January to March. 3 months. All right, so we have 2 answers so far. One of them we had hypotonic because the water is going to move. Well, so the water will move out of the cell. So we have a hypertonic solution, hypertonic, more solute outside of the cell. So those are our 2 answers so far.
Okay.
I don't don't see any more coming in. Maybe we don't have too many January to March birthdays.
That's
Oh,
possible.
we got another one. Hypertonic. The water will move out of the cell. Hypertonic, more solute outside.
Okay, so in terms of how to solve this particular problem, we want to make sure that we look at the solute concentration. So we're looking at the solute concentration here and the solute concentration here. Okay, so where is the most solute concentration going to be located? It's going to be located outside of that cell. So in this case, the water inside that cell is going to go out to the solute in the environment. So in this case, this is going to be considered a hypertonic solution. Good job, guys, for those individuals that got that.
Yeah, we have some more coming in right now. Hypertonic, hypertonic, because the higher concentration of solutes are outside. So wonderful, wonderful job, guys.
Alrighty, next. Same idea. Let's have from April to September. So this is even bigger group. April to September babies. Okay. You're going to draw the picture again. You're going to make sure to tell me about the movement. Okay, so drawing arrows, tell me about the movement. Is it going in the cell? Is it going out of the cell? And what type of solution? Is it hypertonic, hypotonic, or an isotonic solution?
All right, all right. April to September birthdays. Put those descriptions of where the water will move and what type of solution we have in the live chat. Hey, my birthday's in May, so can I answer this one?
Mr. Ewald, you're not a student. I know you want to join in on the fun.
I do.
You're not a student.
But I know the answer.
That's true. I'm
Yeah.
glad you know the answer. I'm glad.
All right. April to September student birthdays. Let us know what you guys got. All right, so we have 2 student answers so far. And what we're seeing is the water will move into the cell because there's a higher solute concentration, and this would make it a hypotonic solution. Water will move into the cell due to the cell having more solute, resulting in a hypotonic solution. Hypotonic, the water will move into the cell. Hypotonic. Hypotonic, as there was more solute inside the cell. Hypotonic.
All right.
Hypotonic, yes.
Good job, guys. Absolutely 100%. We're comparing the solute concentrations, 40 to 45%. We can see that inside the cell there's more solute. So in other words, we're going to have the water from the outside environment come in to the solute. And so this causes a hypotonic solution.
Wonderful, wonderful, guys. Great job.
Alrighty, now question number 7. Question number 7, we know it's an isotonic solution. Okay, how do we know that? We know that because the solute 45%, solute 45%. So the solute concentration is the same. So your question is about the movement of water. So how is the water going to move in an isotonic solution? Okay, so make sure to draw the picture, make sure to draw the arrows, and then in the live chat, let's have everybody answer, okay? So everybody answer about the movement of water for this isotonic solution.
All right, so once again, everybody in the live chat. What are those arrows going to look like? Or arrow, I don't know. So what is the movement of water going to look like in this solution? We know it's isotonic. Mr. Wen told us that. So what's the water going to be doing in this solution? All right. We have 3 answers so far. I know there's more than 3 of you out there right now. 4, 5, 6. The water will go in and out at the same rate. The water will transfer at the same rate. The water will move in and out at the same rate. It's going to move at the same time, same rate. The water will go in and out at the same, the water will move in and out of the cell at the same rate. Awesome.
Yep, that's exactly what we want to make sure that you guys understand about isotonic. Instead of having that single arrow, that isotonic is going to have arrows that both go in and out of that cell. So
Beautiful.
Good job on that.
Beautiful.
Now, our last thing that we want to do is going to be the live egg demonstration. So what I want you to do is copy this chart down, and I'm going to explain to you what we did in this live demonstration. So we took 3 eggs, 1 didn't make it. Let's just say that. 1 didn't make it. Okay? And now we have those 2 eggs that we put it inside a vinegar solution. We left it for 24 hours. Mr. Yarrow, what happens whenever you leave an egg inside vinegar?
So when you leave an egg inside vinegar for at least 24 hours, the shell is going to dissolve, leaving behind a nice little outer membrane on the egg.
Okay, so absolutely. An egg is a great demonstration of that selective permeability that we've been trying to tell you. And so in case of removing that eggshell, now we have that membrane, that thin membrane that's going to allow for certain things to go in and out. And so then we took that egg that has no eggshell and we placed it in water and we placed it in Karo syrup. Now Karo syrup is like if you were to eat waffles, if you were to eat pancakes and you were to pour that syrup on top, same idea. Very, very thick, very, very sugary. So we're going to see what happens as a result of that, okay? So we want to make sure, again, write down the initial mass, 66.5. Write down the initial mass for this one, 62.3. And then we will show you the live demonstration.
All right, so I'm going to give you guys another maybe 15, 30 seconds to write down the initial mass for the egg before it went in the water, the initial mass for the egg before it went in that sugary Karo syrup. I'm going to let you guys write that down, and then I'm going to grab the camera and take you over here and show you what we have. All right, so here we go. Before I take you over there, you get to see me. Hey guys, how we doing? All right, then I'm going to take you over here to Mr. Wen. All right, Mr. Wen is standing in front of, we have our digital scale over here. All right, we're going to take the mass in grams of these different eggs. Here we have our different solutions. And then Mr. Wen is actually holding our first egg. So let me kind of zoom in there a little bit so you can see that bad boy.
And not only that, but what's really cool is that you can see that yolk that's just floating around. That is super cool.
Yeah, I don't know how well you guys can see that, but it is awesome to be able to see that yolk floating around in there.
All right, so we're going to take the mass of this. Now remember, this one was placed in water.
So our final mass for the egg that was placed in water for 24 hours, 86.57 grams. All right, we started with 66.5 grams. That was our initial mass. And after 24 hours in water, we now have a mass of 86.57 grams. All right, so I'll zoom out a little bit. Okay. Mr. Wen is now going to grab my most favorite egg from this experiment. And he's going to show you a little bit about this.
So this one is super duper fragile. And the reason why is because all of the water is basically out of this egg. You can even see the yolk, and I can touch that yolk. It's so weird.
How cool is that?
And you can see the folds because of the fact that there's not much volume left in this egg. So now we're going to mask this.
So we're going to put this on the scale. And our final mass for the egg right there that was in the that sugary Karo syrup for 24 hours, our final mass is 34.34 grams. All right? So our initial mass, sorry, I'm moving around a little bit. Our initial mass was 62.3 grams. and our final mass was 34.33. Now, Mr. Wen, really quickly before you move on, could they do this at home?
Absolutely, you can do this at home, but of course, remember, you have to have your parents' permission. So all you have to do is find a container, find some vinegar, and then place an egg inside that vinegar container. Wait about 24 hours, that eggshell will come off, and it will be super duper cool. Okay, so you're more than welcome to check it out. Real life biology at your home. Alrighty. So if there are any other questions, please let us know through Schoology or Teams, and make sure you let your teachers know as well. Okay, so in terms of make sure to turn in the questions. So I have an assignment already available. I believe that Mr. Ewald has a discussion available for his class, so make sure to turn those questions in. Make sure to check for Schoology for instructions on how to submit it. So if you're not sure, again, ask your teachers. Check Schoology to see whether your teacher has assigned any more work. So I know that I have assigned a virtual lab, so make sure that you jump on Schoology and check that out under the NTI folder. Okay? So that assignment takes about 10 minutes to do. It's not very difficult at all, but again, it kind of highlights a lot of the different points that we try to make. Okay? Mr. Ewald, do you have anything else in terms of assignments?
Yeah, I'm going to hop in here really quickly at the end. So for my guys, for my fourth period, make sure that you hop into our Unit 3 folder on Schoology. You will see the discussion for the live lecture. All right, make sure that you answer those questions completely. Make sure you submit before the end of the day. All right, we do not have anything additional to do just yet. We're going to do that next week after our next live lecture. So all that you guys have to do for Mr. Ewald's class is hop onto Schoology before the end of the day and complete that discussion for the live lecture. All right, more information to come about our additional work next week. Okay, the last thing we want to be aware of, and I'll try to get out of the way so that you guys can see it. Our next meeting, our next time-bound event, is going to be another live lecture on active transport. Okay, so for you guys, I'm pretty sure that's going to be Tuesday, right? I think Tuesday is our next even day. So Tuesday, whenever the next even class period is, we're going to meet again for a live lecture. So thank you guys so much for joining. I'll throw it back to Mr. Wen to finish it up. Good stuff, guys.
All right, guys, so thank you so much again. Just exactly what Ewald said. We really appreciate the participation. We know the NTI has already been a struggle for some of you guys, and that we are here for you guys, and we want to make sure that you guys get your questions answered and you know that you're being supported, okay? So we thank you guys so much. We hope to see you again next time.
You guys are awesome. Enjoy the rest of your Friday. Enjoy the weekend.
Which seat do you want to be in? You be in this seat? Be in this seat. It's already down.